We are at the Salk Institute. We're actually in the computational neurobiology lab. This is the tea room, and we've just come from the stem cell meeting on the Mesa. It's actually the 10th stem cell meeting on the Mesa. And the keynote, the morning keynote, was given by Elaine Fuchs, who's with me today. And she's actually, to get this right, Lancefield Professor of Mammalian Cell Biology and Development and an HHMI, Howard Hughes Medical Institute person at the Rockefeller, at the Rock in New York. And amongst other things, a National Medal of Science winner, which is a significant honor. Your talk was actually titled Stem Cells in Silence, Action and Cancer, which is intriguing. And so could you just elaborate a little bit on what that means?
I think some stem cells in adult tissues spend most of their time either in a resting stage where they're not making tissue. Most tissues don't have that many cells that are dying constantly or turning over constantly. Most of our tissues are doing okay. And so our adult stem cells within those tissues are quite happy, just resting,
right,
not doing much of anything.
That would be called quiescent?
Quiescent, yes.
Okay,
and so
quiescent.
Now you wound the skin, for instance, and all of a sudden you've got immobilized stem cells. They have to respond to that signal. They have to go out, repair the tissue, and they've got to be prepared to repair whatever damage has been done. And once they've done that, they have to go back again, say, okay, the tissue's repaired, go back and become silent again. And so I'm fascinated about how that happens.
Right. So it says here in one of the many things I've read about you, this work on the mammalian skin diseases, hair, and so on, has led to the modernization of dermatology. I've actually seen a couple of pieces here which are from fashion magazines, in fact, and talking about hair and skin and so on. How does this, the whole notion of hair and skin and stem cells being the same stem cells being the source of the, could you elaborate on that? Because it's kind of intriguing.
Yeah, it is intriguing, isn't it? Because we look at our skin surface and then we look at our hair.
Right.
They look like completely different structures,
Yeah.
and yet they both are coming from stem cells. And there are different compartments of stem cells that exist within our skin, but in a wound response, the hair follicle stem cells, which are deeper in the skin than the epidermal stem cells, if you remove the epidermis, which anytime you can see blood on a scratch, you've gone straight through your epidermis. Your epidermis is cellophane thickness.
Right.
And so we typically will remove our epidermis in one way or the other. We do that pretty constantly in terms of the course of our lifetime. And if we didn't have stem cells that were deeper in the skin, then repairing a large wound on our skin would be a really challenging thing to do. But fortunately, we've got multiple different reservoirs of stem cells, some within the hair follicle. And when there's an injury, those stem cells can go up and repair the epidermis in a way that normally they just make hair.
So, 3-year-old boy or girl running down the street, falls over, large scream, and knee is cut and bleeding. What's happening there?
Stem cells in action, 911. So if the boy is bleeding, then clearly his epidermal barrier, which keeps harmful microbes out, essential body fluids in, has been damaged. And if that didn't repair itself, the child would succumb to infection or dehydration. Either way, it would be a very damaging situation. And so in those kinds of cases, in injury cases, there really has to be a response of stem cells. Anytime you see your wounds repairing themselves, that stem cells in action.
Now, the reason I'm asking this is that we've been doing this meeting for 10 years, and a lot of the talks have been at a somewhat rarefied level in terms of stem cells. And they're thought of, I think the public thinks of them as this new discovery, this very recent thing, not what's happening when you fall over and cut your knee. Do you see what I'm talking
exactly,
about here?
Exactly.
So the history of this field, the recency of it, the areas that it's currently looking at, and the hopes that people have for actually being able to use stem cells to repair serious diseases. I mean, this is all extraordinary and fast.
It's very exciting. If you think about the history, though, of stem cells for regenerative medicine, you're really going way back in history. And certainly when I was a graduate student, I heard a lecture by a professor at MIT who had discovered this method of taking a small piece of human skin, putting those cells into culture and expanding those cells and in large fashion, where he could make plates and plates and plates of epidermis under conditions where the epidermis would be able to repair tissue, would be able to still function. And in fact, we didn't call them stem cells back then, but that's what they were. They had cells that had the capacity to be able to churn out tissue for many generations at a time. And my former advisor, when I went to his laboratory to train, my former advisor developed that methodology for the treatment of burn patients. And today, when a badly burned patient goes into a clinic for treatment, most hospitals, most burn trauma units have culture facilities associated with them, and it really dates back to what we have is basically more than 3 decades of success of the use of stem cells in therapy. Analogous to that are cases of industrial blindness, where some industrial chemical will blind someone in one eye. If there's a good eye and good stem cells, it's possible to generate sheets of corneal stem cells that can be used to treat the blindness in the other eye. So again, years of success in these treatments. I think what excites people nowadays is that the basic fundamental methods for culturing epidermal stem cells, which came into play back in the mid-70s, were the same methodology that was then adapted to the culturing of embryonic stem cells. And now with the ability to make induced pluripotent stem cells, all of a sudden the doors open not just to make skin stem cells or to make corneal stem cells, but now any type of stem cell, whether it's neural stem cells or pancreatic beta cells or any of the above. And so I think the doors for regenerative medicine are now open in a much broader way than they were back when I was first starting out in stem cell biology, and that's a good thing.
Yeah. So a lot of these practical issues. I mean, do you get questions from people about men? Can they cure their baldness? Because obviously there's a deficiency of something going on there.
Yes. Well,
I remember Craig Venter followed you at a talk and said that he thought perhaps his stem cells were quiescent on his scalp.
I mean, certainly my husband would have much rather had me go into something practically an applied science as opposed to just understanding the basic mechanisms of how stem cells work. However, actually, in understanding the basic mechanisms of how stem cells work, we discovered a number of years ago now that there is a signaling pathway, which is called the Wnt pathway, that plays a critical role in stimulating hair follicle stem cells to make hair. And it turns out that in male pattern baldness, the mechanism of activating the stem cells doesn't involve the exact same molecules, but the same signaling pathway that we discovered a number of years back. And that turns out to be what's critical in stimulating hair growth in humans, as it was in mouse hair. And it turns out that it's that pathway that is defective downstream in male pattern baldness. There's a specific gene upstream that is implicated in male pattern baldness, but it all funnels into basically the same fundamental mechanisms. So I think even though my laboratory will continue to study the basic mechanisms of what turns stem cells on and what turns stem cells off,
right,
we're delighted to see that there are many people that are interested in a billion-dollar industry of solving the problems of baldness.
Is it? But there isn't a solution yet. I mean, there's lots of products out there.
Lots of products out, and lots of things that stimulate
Not that
stem
I would
cells.
know. I mean,
yes, right. Well, you and I don't have too much to worry about, but basically there certainly are a number of people that are very concerned about this. And I think one of the interesting aspects and one of the challenges that we have is the question of how potent are stem cells? How much can they be used before they exhaust themselves? And for instance, we worked out a method, we worked out a series of different genes that are involved in activating the stem cells. And we can overactivate the stem cells when we want. In fact, in aging, our hairs get sparser as we age. We understand that very clearly in terms of why that is. There are a series of proteins that promote the stem cells to just be silent, and we can stimulate those in mice. It's very easy to do. We can stimulate hair growth. What we don't know is, and those hair follicles were otherwise just happy sitting there, never doing anything in the aging mouse. So we can stimulate the hair growth.
Right.
But now the question is, now that we know how to stimulate the hair growth, we have to monitor this over an extended period of time. Because what happens if there's a time limit on the number of times you can activate a stem cell? So we activate the stem cells, get a lot of hair. Now, what happens after a year or so? If those animals end up losing all stem cell capacity, they'll lose all of their hair, and people wouldn't be very happy with us in terms of rushing to a clinical trial or an applied trial to see this kind of activation in action. So I think those are the challenges that we and others have in terms of stem cell biology, and it's not at all clear, even though we think of stem cells as a fountain of youth, and embryonic stem cells certainly are.
Right.
Whether adult stem cells have this fountain of youth capacity to just endlessly churn out tissue is something that there's very little known about the answer to that for most tissue types. So I think those are ongoing questions in the stem cell field, and clearly for the future of regenerative medicine, we really have to understand the answers to those kinds of questions.
Yeah, I think the term regenerative medicine or functional medicine, these are fairly recent terms really, which have grown out of this whole area, I suppose.
Yeah.
In terms of exactly what you can do in terms of these repairs.
Well, I think the excitement is that stem cells in wound repair often have a greater repertoire of the types of tissues that they can repair than they do in their normal setting. Normally, they're sitting in a niche, in a protected niche, and the niche is telling them, do this, do that, be quiescent, just make hair. But in a wound setting, our years of evolution have basically allowed every tissue of our body just about to really send out these 911 calls to stem cells, where stem cells in the vicinity, even though they normally don't make a particular tissue type, will do so if they have the capacity to do so. So, for instance, the epidermis and the hair follicle stem cells can make epidermis, sebaceous glands, to some extent sweat glands, but they can't make, unfortunately, neurons or muscle cells or pancreatic islet cells unless we reprogram them to do so, to this induced pluripotent state. But normally, when they're sitting there in the skin, they can do these things. And I think that's the word that's being used in the stem cell field now is called plasticity. It's the ability to do things that they normally don't do. And I think for regenerative medicine, it's actually a very good thing. If we understood enough about what kinds of instructions stem cells naturally go through as they're being instructed to make tissue or don't make tissue, because don't make tissue is just as important as making tissue when you think about it.
Right.
You don't expect you expect your wounds to heal, but you don't expect to get tumors where your wound once was. So we have mechanisms to turn it off, and those are as important as turning them on. And I think the more we understand about those light switches that are turning stem cells on and off, the more we are going to be able to contribute to critical aspects of regenerative medicine in many different areas.
When did you have the first sentiment, the first clue that you wanted to go into this sort of area? I mean, not even this area, into science at all. I mean, what's the beginnings of all of this? So it's usually a parents or a book or a teacher or something. But you had family connections in science.
Well, I did have family connections in science. My father was a geochemist. He worked on meteorites, looking for extraterrestrial minerals that didn't exist on Earth. And so during the lunar landings, he analyzed the lunar samples. He said they were very uninteresting. They're too close to Earth. But these meteorites that come from outer space and fall wherever they may fall, those were fascinating to him. And
this was Argonne National Laboratory.
And this was Argonne National Labs. And he was essentially a one-man show there. Not that many people studied his field. I think when he retired at the time, there were something like 13 extraterrestrial minerals that had been identified, and he discovered 8 of them. The one that I most distinctly remember was one that he had named after a Russian geochemist back during the Cold War. And so as a child, receiving when the family received Christmas cards from a Russian scientist during the Cold War, that was just wonderful. It was just maybe a little bit of an icebreaker. And my aunt, his sister, was a biologist, a technician at Argonne National Labs, and she always wanted to go to medical school, and her grades were much better than my dad's, but she couldn't she couldn't get into medical school. That was during an era where very, very few women were ever admitted into medical school, and so she was resigned to being a technician, a biologist, and telling her nieces that they can do whatever they want to do, and please do it for the sake of not only what you're interested in doing, but also for the sake of women. And it was good advice.
I did mention that this morning when I was introducing you. I noted I had a bunch of your papers to hand. The latest Nature paper, Pioneer Factors Govern Super Enhancer Dynamics in Stem Cell Plasticity and Lineage Choice. I had the nice, very good review summary that you did with Cédric Blancpain.
Yes.
On stem cell plasticity the year before. I had the SOX9 paper. And then I dropped this other one, which is also 2015. 7 actionable strategies for advancing women in science, engineering, and medicine. And some of these other names, of course, Fiona Watt and others are well known. Why did you feel it necessary to write this?
To participate. Maybe part of it is inspired by my aunt, who was a staunch feminist.
Because you've been quite outspoken on women in science and issues.
And I think that her experience has, and her outspokenness for what she believed in, not simply women's issues, although women's issues were very important to her, but she was willing to speak out and on whatever plane that she felt was important for her to speak out about. She was not shy, and in fact felt that the right thing to do is to be vocal when you see something happening that you don't feel is right. And so I think she definitely influenced me tremendously along these lines. I think in fact, much of my early career, I was not concerned at all. In fact, I remember her once asking me at a dinner that we were at when she came to visit in Chicago. She said, well, have you ever felt discriminated against in science? And I said, oh, me? No, not at all. And we started, and she said, well, that's odd. Your sister has. My sister is 4 years older. And I thought, no, I've been fortunate. I've escaped that. And then I started reflecting back and realizing that that in fact I worked. My graduate advisor had said that women didn't belong in science, and to me it was just a challenge, saying, well, I guess I have to do that much better to be able to make sure that he does feel that I belong in science and I belong in his laboratory. And there are many examples of that where I hadn't even thought about it. Just took it as a challenge, feeling that I need to do better. And so I actually don't want to lose that challenge. I don't want to let that go, because I think we can all do better all of our lives, and I do think that I can do better, but I do also want to make sure that women who are coming up through the ranks don't face some of the situations that I and my fellow women colleagues have faced over the years. And unfortunately, it's not entirely disappeared in our culture.
Yeah, my colleague Claudia Dreyfus at New York Times, the conversation you did with her, you said you remember as one of 3 females among 200 male chemistry majors at the University of Illinois. I was terrified that if I did well in class, the professors would think I'd cheated.
Yes.
That's how much I didn't think I belonged. So I studied like crazy and routinely got the best grades on examinations, because if I was number 1, then who could I have cheated from?
Exactly.
So you covered yourself. It seems to be getting better.
There's more than 3 women now in a class of 200 physics majors.
Yeah.
But it does seem to be getting better. But on the other hand, we have typically, one sees in graduate school, sometimes 50% of the graduate students are women. And so you say, with those kinds of statistics, things are going to be changing very fast.
Yeah.
But in fact, when you look at the number of women who are going up through the ranks, when you look at the top, there are very few indeed. And when you look even at the assistant professor level, it's far from 50%.
Yeah.
And in fact, far from 50%. And so it's not that every woman is going to necessarily go into science or feel that science is what she wants. But I think it's important that the nuances that are out there disappear. My colleague Shirley Tillman said that if you look at if you ask a man to name the top 3 scientists in a particular area, that typically he will think of 3 men.
Yeah.
And if you ask a woman to name the top 3 scientists, she may not always choose 3 women, but the chances that she does choose at least some women, if not 3 women, are much higher than they are for the man. And I think it's that perception. If you have at the top 20 men in a particular department,
Right.
and you recognize the need to hire women, that nuance of how you perceive the outstanding scientists, just the subtleties of not even thinking about this person's science or that person's science and including that as being an outstanding contribution of work, becomes a problem. And I don't think it's not a conscious problem, because you ask the same male colleagues and they just honestly don't think that that's the case. And so I think it's a subtlety that still needs a little bit of work.
So what would be, there was a little book by Sir Peter Medawar years ago called Advice to a Young Scientist. You've had plenty of people go through your lab now. I mean, you can focus this on women specifically if you want to, but what is your advice to a young scientist?
Yeah.
What's ahead of them, how to best navigate?
Yeah, I mean, my advice, certainly the first advice, is definitely to pursue questions that you're passionate about and not to think about the future too much. The future is going to fall however it's going to fall. I think when I was a child growing up, it was the sort of Doris Day, que sera, sera. But there's a little bit of that to it. I mean, you can do as much as you can do, but be the best you can, and you've got a better chance of the que sera, sera. But
it's Taylor Swift now with the guys.
Yes,
yeah, right,
I'm certainly out of touch these days. But I think it's that aspect where if you're passionate about a question, and even if you're not trained in that particular area, you get excited. For me, I was a graduate student in the biochemistry of bacterial sporulation when I heard Howard Green speak, and I was so wrapped with his system of growing skin. I thought, that's what I want to do. And I think back, if I didn't follow that instinct, even though I had never worked with human material before, I had never done cell culture before, I wasn't thinking like a cell biologist. Had I not followed what I was really excited by, I'm not sure I'd still be as excited as I am today about the science that we do.
What's your take on, I mean, you travel a lot, you get invited to places, you go and talk and so on, you just got back from Vienna. What's your take on the state of the relationships between science and the larger public at this point? I mean, it's very nice that there were 2 Academy Award nominees for playing one Stephen Hawking, one Alan Turing,
yes.
one won an Oscar.
Yeah.
Very nice that my friend Neil Tyson has his talk show. Lots of good stuff going on in science, but there's also all these retractions. There's the irreproducibility, there's the peer review issues, there's the Pew report talking about the lack of public trust in scientists and so on, and science now. I mean, so there's this best of times, worst of times things going on.
Yes.
What's your sense as you go around?
My sense is probably not much different than the general problems that we have in our society today, and I think it gets at the core of 99% of the people out there are doing science in a rigorous way, in the ways in which they were taught, in the ways in which they know is right. And then there are the pressures that exist in society where those odd people out are succumbing to a pressure of publication in the top journals, all of these things that instead of working to get those publications, and sometimes, you know, science is up and down. We don't know what nature's going to hold under her hand of poker. So sometimes the cards come up and it's not what you expected, and it's not interesting. And as a scientist, those of us who still work and love science have gotten used to the ups and the downs.
Right.
Have gotten used to spending a year's research working on an artifact. We're still excited by science. But we don't step that boundary. And the fact that some people do step that boundary, it's nothing new. It's happened back when I was a student. There were these cases of horror stories about these kinds of things, and yet I think certainly it's gotten more visibility, but it's not so much. It's certainly not the norm of the science that I know my colleagues do, or the science that their people do. It's a relatively small percentage of people that I think have go in with fraudulent intent to manipulate data. And in fact, when you look at science, science is, if it's exciting, if what you've done is exciting, there are 10 people out there wanting to repeat what you've done and move on.
Yeah.
And so if there's a problem, these problems surface quite quickly.
Yeah.
And so you have to ask about the mindset of saying, it's so obvious that the problem is going to surface. Scientists will catch the errors. If it's not reproducible, science will catch that. And if a few people doubt it initially, but it turns out to be right, science will catch that too.
Right.
It may take a little bit longer as the pendulum swings, but basically, I think the truth in science does come out, and so to have a few, whether they recognize that that's going to happen and they just are doing it for the moment, or whether they really are so far out of touch that they don't recognize that that's happening, it's a tragedy. And yet it's one that I think all fields have.
Yeah. If we're actually sitting at the tea table in the lab here, but which makes me think of a dinner table. If I gave you a time travel token, so that you could bring to the dinner table anybody from any discipline, from any time of history, do you have somebody you'd really passionately love to sit down and ask questions of?
Now, well, does it? Do I have to choose a scientist?
No. anything at all.
So, I mean, I would say that I'd like to find out about Abraham Lincoln. So he was somebody who both my father and my aunt had his picture in their study.
Really?
They were both staunch Democrats, and they both brought me. My father brought me through the poorest neighborhoods of Chicago, saying, "I want you to recognize that you." That not everyone is as fortunate as you are, and that had an impact on me, and yet Abraham Lincoln, in his efforts to abolish slavery, had to make compromises, big compromises, in the same way that political issues now play themselves out in Congress, and I think it's interesting that a man who had done so many positive things for our country also had to deal with politics, with persuading people in ways that sometimes perhaps weren't as pleasant as others. And I think those conflicts of great men are things that I think we would, I would like to know more about. I could go on because there are also great women in Marie, Madame Curie.
Of course.
I'd love to have a conversation with her. There was a conversation that I had with Barbara McClintock when I was a graduate student.
Oh, right.
And she was an inspiration. I remember asking her, well, how did you feel when they wouldn't even give you a laboratory at Cold Spring Harbor? And she said, I had my corner where I had my microscope and I could do my experiments, and I was thrilled.
Yeah.
And I really, as someone who has resonated to that, saying, no matter what out there, if you follow what you're really interested in and you do it with rigorous science and you do it with the right standards, then people like her will change the view, and as well as do some spectacular work that changes the face of science, that changes dogma, that challenges dogma.
Yeah.
So there's and I guess all of I guess I would love to have a time warp to just walk my way through and really have conversations with. We could fill this table with all sorts of interesting individuals.
Sounds like a somewhat political household you grew up as, politically aware household. Because you marched, didn't you? And there was a Vietnam protests, Peace Corps stuff, and so on and so forth. So you were an engaged teenager and young woman at that point, right?
Yeah. My father was always saying, well, I don't mind that you're marching. I think that's a good thing. You're doing what you think is right. But would you mind being in the back? I think he was always afraid that something was going to happen. So, it was that sort of fatherly love, but on the other hand, recognizing that this was important. So, I think I cherish the fact that I had a family who is mindful of the importance of the world out there, not simply one's own egocentric self, which we're always so immersed with ourselves on. And my dad was definitely one of those kinds of people that would take up conversations with the gas attendant, gasoline attendant, whatever, and he make everybody feel comfortable, and it's such a good role model to have parents that have that and that hold those things so importantly to themselves.
There's the Lincoln thing is interesting as well. There was, and this is just an aside, but there's an extraordinary book by Gary Wills on the Gettysburg Address, in which he analyzes it down to the last word. But he also looks at the first inaugural, second inaugural as well, and he shows the original version that Seward had written, and then Lincoln's version of it. And it was amazing to see the difference between, although the skeleton is there. These wonderful words were written by Seward, but then Lincoln comes along and just puts this beautiful
touches to it,
which is just extraordinary. So the reason I mentioned that was that in terms of writing, I mean, do you have any plans have you any ambitions to do any writing as such? Beyond the science paper?
Yeah. So right now, I do like writing reviews. I have to say my favorite is to write manuscripts of the latest data, to work on that. I mean, obviously I ask my students, I ask my postdocs to write the first draft, but I love to put touches on on the writing. I enjoy writing review articles that have something interesting to say. And my husband always says, well, of course you're going to write your memoirs. And I say, well, of course, when I'm not so passionate about my science anymore. And he says, when is that going to happen? And I said, well, I'm not quite so sure about that.
It's the same material.
Yeah, right. In a way, it is the same material.
Yeah.
And I think it is important to capture the history of science, the history of the fields that each of us have been in for a number of years. I think that perspective I enjoy having, whether it's writing an article and putting that perspective of more than 3 decades of experience towards, or whether it is writing something broader from a historical perspective.
Right. So they're going to have to get ready for tea very shortly. It's a different person in the lab does it every day, so they'll be getting ready. So let me ask you just the final question. A couple of questions here. One is, where do you see the stem cell field most likely going next? And I raised an issue in there as I introduced the afternoon session before I came over here, which is that along with the new CRISPR-Cas9 gene drives, gene editing technologies, and so on, new ethical questions are arising. Again, Jennifer Doudna, so there's another woman at the forefront of these sort of things. When we started the stem cell meeting on the Mesa 10 years ago, we had ethics panels for the first 2, 3, 4 years.
Yes.
And then it was all talked out. And people were happy. There was a good discussion. It became less important to have to deal with that every meeting. I'm beginning to wonder if it's going to start coming back again now as we get more advanced gene editing technologies going on. In England, the Crick Institute that asked for a government permit to do some work on embryos and so on and so forth.
Yeah.
So, with that as an aside, I mean, where do you see the field going? And
certainly, with regards to ethical issues, chimeras between human cells and animal cells, that's something at the forefront, because when does the animal become too human, and are we going to give the mice a human brain? Those kinds of issues, I think, are definitely ones that merit revisiting of ethics. With regards to CRISPR-Cas, you could argue the same sorts of things. Some of the genetic skin disorders that my laboratory worked out over the years can now easily be corrected, I think, with CRISPR-Cas technology. And I don't think anybody, or very few people, will have an ethical issue about correcting a devastating disease. And if you already have families that are not, if there's a 50-50 chance of their child having a devastating, life-threatening autosomal dominant disease, then those parents are already going through in vitro fertilization, trying to check which of the eggs are healthy, which ones aren't. That's been going on for quite a few years. And so now, when the technology exists to correct them in a much faster and much more streamlined and much safer way, I think those are going to be the first types of CRISPR-Cas technologies applied to the clinic, and no one's going to have a problem with that. The problems arise with additional types of CRISPR-Cas technology, and when are we going to stop at the level of blue eyes versus the level of a basketball player, another foot, all of these kinds of issues. Obviously, then there are lines drawn, but like every field out there, there are dangers and we as a society come together and we decide where we cross the line. And I think scientists are no different in that regard. It's important of people being cognizant of the limits, but it's not differently than, say, people using utensils in a kitchen, which could be very dangerous, but if you're using it to cook a souffle, I don't think anybody has a problem with that.
It's a scale issue, I think. But,
Exactly.
but you're obviously aware of this, because as I read this morning, I also read this passage from your 2000 commencement address at Chicago. I now balance the joy of discovery with the necessity of taking seriously ethical and educational concerns at the nexus of science and society today. And of course, you know, science and society is a very major concern of ours at the Science Network, the Science Collaboratory. Indeed, for the world of science to be a successful one, it must be a science of the world. It must be a science that embodies concern for the world of the next millennium. Your education, students, has taught you to be morally and ethically responsible and to bring philosophical reflection into your chosen profession, your community, and your life as a whole. So you obviously have this resonances, which sounds to me like that was part of the description of your father and your mother as well.
Yes,
really instilling this into you.
Yes, father and husband. My husband is a director. Yeah, he's a director of philosophy and education at Columbia University Teachers College. So, so our commonality is in education and philosophy, and I think that that has only strengthened what my parents have taught and my aunt and uncle have taught. And so, again, I feel that many different types of science, the environment first and foremost, I would say at this juncture on our planet, we as scientists have a responsibility to educate the public, not just to sit in our ivory towers. And as much as I love to do science and love to do experiments, it's important to be out there. It's important to explain what it is that we do, and it's important to do so in a way that that the public can understand what it is that we're trying to say. It's not so difficult to explain what you do if you think about it, and if you're only used to talking to other scientists, then and you don't take the time to think about the public, then all of us are at a loss for that. And so I think that it's our onus. We really have to go out there. We have to participate much more in scientific community, in the community at large, in the world at large, and we can do it. We have an immense talent and immense passion. We just have to apply a little of it in these right directions, and it's going to be important for the future of the world.
What are you optimistic about? That was the last question.
Oh, I'm optimistic about regenerative medicine, about treatments, about cancer therapy. I have never seen so many different successful means of attack and multiple fronts of attack scientifically, with good, strong, basic science founding, that is now really starting to, I think, make a difference. And if I could go out there now and convince Congress, convince the public that that 40-year war that has been going on in cancer, people have heard it. Yes, it's right around the door, and then it's not around the door, and I think there have been so many excitements and so many disappointments, but I cannot help from being really excited in this era. I've begun to really see a change in the way in which scientists interface with translational medicine. The language barrier is no longer what it was when I was a student. There was this big wall between medicine and science, between medicine and technology, between medicine and pharma, and biology and pharma and medicine. all the various different disciplines. Every one was an island. And now people are working together in very effective and very creative ways. And I'm very excited about the future of translational medicine.
All right, well, that's, I agree with you. I see the same sorts of things. It's literally in the last decade.
Yeah.
It's just amazing. And the commentaries that were coming out of the partnering meeting for the last 2 days, industry sees this as well. It's just quite remarkable. So thank you very much for joining us. Thank you for sharing. Thank you for giving the keynote.
My pleasure.
Elaine Fuchs.
Thank
Thank
you.
you very much.